on biogeochemical cycling and microbial ecology. So microbial ecology is just looking at community dynamics and interactions of microbes with each other, plants, animals, and the environment. So it's pretty much similar to animal ecology for that matter. The cool part is that microbes play a major role in life on Earth, and the downside is we've only cultured maybe less than 1% of all the species, and so it creates a big issue when you're trying to figure out who's doing what in the environment, or even who's doing what in your gut tract. But we're developing tools to really ascertain this information and try to understand it better, as I'll talk about. Some of the foundations of microbial ecology, no different than plant or animal ecology. Populations are assemblages of similar organisms, usually assemblages of the same species. Communities are mixtures of different populations. And ecosystems are self-regulating biological communities in their physical environment. And so again, the interaction of these microbes with each other, with other microbes, and the interrelationship between those biological communities and everything that's around them is the ecosystem concept. Biogeochemical cycling specifically involves biological and chemical processes, and so it's a lot of redox reactions. How microbes are cycling nutrients through the environment, and again, detoxifying things or adding certain nutrients, or even taking some away. And so the first main cycle to look at is the carbon cycle. And obviously, carbon can come in many forms. There's reduced forms like methane and organic matter, again, like glucose. And then oxidized forms like carbon monoxide and carbon dioxide. So this figure illustrates the carbon cycle. On the right is the aerobic processes, and on the left are the anaerobic processes. And so we start in the center with some kind of organic carbon, so again, some sugar or whatever. And microbes can respire that sugar. Again, and I've gone through oxidative respiration, showing you the role of glycolysis and Krebs cycle and electron transport chain in that process. And so in the process of respiration, as the microbes are, for example, decarboxylating pyruvate, they're belching out carbon dioxide. As the microbes are using their TCA cycle, they're belching out even more carbon dioxide. But that carbon dioxide isn't just a waste product that builds up. There are microorganisms, particularly aerobic photosynthetic microbes, that can fix that carbon dioxide back into organic matter. So a photosynthetic microbe might take that CO2 along with sunlight and fix that into organic matter. I'll get into that down the road. The organic matter can also be anaerobically respired and or fermented by many microorganisms. And so in that particular case, you're still putting out CO2, whether it's fermentation or anaerobic respiration. And then the CO2 can be fixed by anaerobic photosynthetic microbes. And so again, back to organic matter. And again, it doesn't have to be photosynthetic microbes. I'm just giving you an example but carbon fixation can occur by many kinds of microbes, not just photosynthetic microbes. So the microbes can also produce an abundance of hydrogen. And so that combination of CO2 and hydrogen can allow methanogens. So these are archaea typically, not bacteria, but archaea, that can take that simple carbon and reduce it to methane. But again, methane is not just this waste product, and it's thankful. I mean, obviously, humans, we use the methane as natural gas to, as an energy source, but microorganisms can also use this methane as an energy source. Remember that natural gas is highly explosive, so you don't want it building up in the environment anyway. But if the natural gas or methane percolates into the aerobic zone, then methane oxidizing bacteria can grab a hold of it and convert it into CO2. And so here's a chemical profile of some sediment, freshwater sediment. And so in this particular case, the microorganisms are, and it could be brackish water too, it doesn't have to be fresh water, but generally this is, and you can think of this as like a swamp, for example. And so in this particular case, you have a water column, and then an oxic-anoxic interface, and then some mucky sediment. And deep below the sediment is an anaerobic zone. And so over here you see the gradients of different compounds, so oxygen and polysaccharide and methane. And so, not surprisingly, oxygen is abundant at the surface and then gradually diminishes as you get to the oxic-anoxic interface. There's a polysaccharide gradient as well. Imagine some things are floating in the water column and it gets a little bit narrower as you get closer to that interface. Down below, however, there's lots of methane, and its levels dissipate as you get closer to the aerobic zone. So in this particular case, way down here, the methanogens are doing their job. They're grabbing CO2 that percolates down from these other organisms, and any abundance of hydrogen that's available, they can reduce that CO2 to methane. The methane can then percolate up, and then methane oxidizers in the aerobic zone can grab onto that and once again oxidize that to CO2. And then over here, the fermenters can take polysaccharides at this interface and ferment those to CO2 as well. And so in either case, CO2 is provided for the methanogens, and the methanogens in turn provide methane back to these groups of organisms. Over here is a picture of the something that we used to do when I would do research out in Massachusetts. There was a swamp out there, and we would stir up the methane and collect it and then light it on fire. And so that's what you see here. So if you get a chance, do a Google search for Volta experiment just to see that process. So the degradation of organic matter is influenced by several factors, and obviously the types of nutrients present in the environment is important. Again, microbes are only capable of eating the things that they have the genetic capability of doing so. The abiotic conditions are important as well, such as temperature and salinity and pressure and all those other things. And obviously the types of microbes present really matters as to whether or not organic matter will degrade rapidly or persist in the environment for long periods of time. And these are basic concepts, but it's really important to think about, because if you have a situation where you're trying to degrade some kind of toxins in the environment, these are all the things that you have to think about. Sometimes microbes aren't going to want to eat oil, for example, or jet fuel or whatever, unless there's other nutrients present. And if the abiotic conditions and the right microbes aren't present, and if the right microbes aren't present, sometimes you can just add them. So this table looks at a couple other different concepts, one of which is the substrate characteristics. And so the main things to look at is over here, the substrate of starch, for example, that could come from, if you chuck some starchy vegetable, potato or something into the water, or chuck it into the woods or wherever some starchy thing might come from, some natural thing that came from a tree. But the basic subunit is glucose, and the linkage is an alpha 1,4 linkage. So the alpha is typically associated with energy sources. And so degradation can occur both with and without oxygen. Cellulose is abundant in the environment as well. It obviously comes from plants and things. And again, the subunit is glucose, but this is a beta 1,4 linkage. And again, celluloses are more structural, and so this more rigid beta 1,4 structure, it helps maintain structural integrity. So again, these things can also be degraded with and without oxygen by certain cellulolytic microbes. And there's another one, hemicellulose and lignin, which is found in wood, and chitin, which is found in lots of different insects, the exoskeleton of grasshoppers and so forth. Proteins that obviously consist of amino acids and are bound together by peptide bonds. Lipids, or glycerols and fatty acids, and again, the bond linkages are ester or ether linkages, as I've shown you. And certainly nucleic acids are a big polymer in the environment that microbes can break down as well. The process of mineralization is the decomposition of organic matter to simpler inorganic compounds. So if you take something that's like a organic protein, so like from a dead animal, and that those proteins are mineralized to ammonia. Nutrient immobilization is when microbes basically will take a nitrogen source, say like ammonia, for example, and then bring that into the cell and then incorporate that into amino acids. And so that nitrogen that got incorporated into an amino acid is now immobilized for a while and unavailable to other organisms until you die and release that nitrogen. And so that brings me to the nitrogen cycle.